All right, now we're going to look at tonicity, what it is and how it's effective in biology. So first off, when we look at tonicity, it involves two separate solutions that are separated by some kind of semi-permeable membrane. So this is a perfect example, a classic, where you have a quote-unquote red blood cell, white blood cells, you can choose whatever you want, that's present in some kind of solution. So, and that solution, the difference between these two solutions is the different concentration of the solute. All right, we have the different terms that we're going to be looking at, different terms of concentration and how they allow for the mobility of water through these different membranes, right? So when we said semi-permeable, what does semi-permeable mean? Semi-permeable means only selectively permeable to specific substrate. In this case, the selectively permeable substrate is water, all right? And so when we look at the different types of tonicities, is we have to look at the concentration of these two solutions in relationship with each other. So when we say that the concentration of A is equal to the concentration of B, so there's a similar concentration. What kind of tonicity do we present in this? This is referred to as isotonic. So iso means the same, right? So isotonic solutions, whenever there is an equal distribution of solute in the two solutions that's present. All right. Now what happens when there's a difference? There's a difference between the two. So now we look at, for example, if A is greater than B. When we look at A is greater than B, what are we looking at specifically here? So A is inside the cell, B is outside of the cell. When you look at terming these, when there is a higher solute concentration of the cell in relationship to the solution, we call this hypotonic solution. In a hypotonic solution, this is where the solution solute is much less than the actual cell. So what happens in terms of concentration, there is an imbalance. And so in order to maintain this imbalance, there's a process of osmosis that takes place. What is osmosis? Osmosis, let's write this down here, is the movement of water from a high concentration to a low concentration. So the diffusion of water that's present. So in a hypotonic solution, what do you expect to see? You expect to see, what am I asking for specifically? The cell. So if it's going to be a hypotonic solution, the movement of water will be in the direction of the cell. So in a hypotonic solution, the cell will quote unquote swell. Right? So in a hypotonic solution, where the concentration of the solute of A is greater than B, this is considered hypotonic. Vice versa, if A is less than B, when we're looking at concentration of solute. So remember that. So I'm going to make a little brackets around these. This is referring to concentration of a solute. What's an example of a solute? Solute is something that dissolves in a solvent. So examples of solute could be salts, minerals. You can have the sugars, fats, protein, anything that dissolves. So this concentration. So whenever now the concentration of solute is less inside the cell as compared to the outside, we call this a hypertonic solution. All right, so this is considered a hypertonic solution. So if we have a hypertonic solution, that means there's more solute in the solution rather than the cell. So what happens to this cell specifically? The cell begins to shrivel up and shrink. Okay, so these are the three terms that we use to distinguish the different types of tonicity in two separate solutions separated by a semi-permeable membrane.